My second year.
It's okay. Now you can speak it now.
Yeah. It's not a core course.
Yeah. You're neuroscience, right? Yeah. CSP. Oh, CSP minor or major? Oh, double major.
Thank you. If you guys are okay with it, just going to take
neuro
through
major
a couple of things
and
real
with
quickly.
math minor.
You'll get, like next week with Dr. Pressy, you'll get the full sort of assignment guideline, rubric, and then the actual workshop related to like how to do a journal presentation. One aspect that I want you to really focus a lot on is that this is not a high impact journal. It's a very interesting sort of result. And there are also things that I want you to think about as neuroscience students as we go through them. Just remember, you're going to be presenting wherever you're going next, and in this course, you're going to be presenting to other fourth-year neuroscience students. So, do you have to like explain like every last detail, or can you make some assumptions about what other your peers will already know? Okay, so. I'm going to talk to you a little bit about this paper. It's not a really high impact paper, but it's a very interesting paper. And then I'll talk to you and ask you a little bit about it at the end. So this molecule that they're kind of looking at, this P4H-TM, it's a transmembrane. The TM part is a transmembrane, and it's actually inversely related to the hypoxia-inducible factors. So if you have a lot of oxygen around, this factor comes into play and actually reduces the level of HIF, or hypoxia-inducible factor. There are a number of isoforms for this particular group of molecules that kind of interact with HIF, and P4H-TM is actually one of those that is found in an unusual place. Normally it's found within the nucleus, but this is actually found within the endoplasmic reticulum of the cells that it's normally expressed in. They are also expressed within neurons, and within the neurons that it is expressed within, it's the neurons that we find the highest levels of this particular factor are ones that are associated with mood as well as to emotional regulation. So it's an interesting molecule potentially. And what I'm trying to do is to get you to understand why this paper might be of some importance when we're thinking about mood disorders. So if you have deficient mice where we have reduced the levels of P4H, they actually have little anxiety. So they don't actually have the same sort of anxiety-related behaviors that you would expect to find in wild-type mice. Okay, so compared to wild-type, reducing the levels of this molecule that is found within these different brain regions seems to have an impact on anxiety-related behaviors and increases their social sort of connectedness. So this means that this potentially could be a really interesting drug therapeutic molecule if we were able to manipulate the expression of this molecule up or down. All right, so do you have any questions on this before I go on? No? So here's one of the many, many different figures that I've actually pulled from this particular paper. So they're using XGAL staining, and wherever you see blue, they have taken a slice of the brain. They have used a transgenic animal where they have beta-galactosidase, and they are staining for the expression of beta-galactosidase where they have P4H-TM. And the areas that you see here, can you name any of them? I think you have the sort of long form underneath anyways, but when it says AMG, can you think of what that might be based on like this mouse brain section? Amygdala. So this is the insular cortex, the cortex here at the top, and the darker the blue color is, the more of this P4H that it has. So what did you learn about in all of your neuroscience courses related to the amygdala? Do you remember anything about the amygdala? You must have heard it at least 50 different times by now. What is it responsible for? So fear conditioning, anxiety-related behaviors, and other things. And what this is suggesting, at least on this slide here on the left, that using this sort of transgenic animal where they have beta-galactosidase as the reporter, the amygdala, the insular cortex, and even some regions of the striatum here at the LS, they have very high levels of this P4H, and other areas don't. So you can see that there are clear areas as well. And again, the lateral septum, the insular cortex, and some other nuclei within the hypothalamus have very, very high levels of this particular P4H transmembrane protein that is inversely related to hypoxia. But surprisingly, if we start looking at these areas, these are the areas that we would traditionally associate with like anxiety, fear-related behavior, etc. Okay, so here's their rationale that we see high levels here in these different brain regions, and it might be of interest to go in and examine this. Now, you told me earlier that the amygdala is an area of the brain that's often associated with anxiety. So can you translate this slide for me? So what are we looking at here? So everyone, it also gives me an idea of like how much neuro you still remember. So what was one of those types of behavioral tests that we do for anxiety-related behavior? Do you remember what it was called?
I think it's like kind of like these two long arms, and then
Two
one
long
of them,
arms.
sorry,
Okay.
they're like platforms, I guess.
Okay.
Across, and then two of them are kind of covered up with walls, and the two of them are just open, and they see which arms of the mice refers to.
Right, so does anyone remember what that's called? Elevated maze. Almost, so you have to kind of combine it together. Yes, at the back. So remember the elevated plus maze for anxiety and fear-related behavior, and the longer you're spending time in the open arms where it's not closed off, the more relaxed the mouse is. And what we're looking at here is the wild-type mice compared to this knockout animal, or the null mice, where we have reduced the expression of P4H. So here again, if we take a look at different ages, 2.5 months after birth, 5 months and 14 months, you can see that there is the beginnings of a change in the behavior. So it's not clear here, it's not statistically relevant, but by 5 months, they are spending more time in the open arms. They are less anxious overall. And by the time we reach 14 months, this separation is cleaner and even more pronounced. So by knocking out this molecule that we just showed to be highly expressed in areas of the brain like the insular cortex and in the amygdala, which is normally associated with fear-related behavior, do they see a positive effect? Do they see an effect on anxiety? I think here, at least on this graph, that it's pretty clear that they are showing an effect related to anxiety. So again, if we take a look, the other aspect of this was not only anxiety-related behavior, but also increased social ability. So knocking out P4H has been implicated in increased socialization. And if we knock out P4H in those areas of the brain, could we actually see that there is increased social ability? So again, we're looking at two different graphs. So the ones on the left are the wild type, both male and female. So they're looking at differences in socialization between male and female. And here, what they're looking at is exploration time. So you either have a cage where there's no mouse, or you have a cage with a mouse in it. And will you explore that cage that has the mouse in it more compared to the empty cage? And if you have knocked out that P4H in those areas that I mentioned before, that overall there is an increase in exploration into the cage that has another mouse in it. So they explore that cage with a mouse in it much more often, so they're more social. And they also spend more time in terms of the social interaction by knocking out this P4H molecule. Does that make sense to everyone? So regardless of whether we're looking at males or females, we see a clear effect by knocking out P4H in these areas of the brain. There's an increased socialization. Okay, now here's where you come back and tell me what you remember. Do you remember the forced swim test? And do you remember tail suspension? What do we use those as an indicator for?
Depression.
Depression. So another type of mood disorder. And this is immobility time. Just curious, because this is where I get to go back and quiz you a little bit. How long do you actually do the immobility test for? Do you remember? Maybe from some of your other courses? Like you just don't keep them up there indefinitely, right? That's actually against all of the sort of ethical protocols. How long do you keep them up there in, suspended by their tails for maximum? So most protocols stop after 6 minutes. Beyond 6 minutes, you actually don't do that. And what you're seeing is, if you're suspending them by their tail, how long do they stay still? So they're not struggling anymore. And do you see a clear difference where you have knocked out P4H, that they continue to struggle? So during the entire duration, let's say 6 minutes, there's never a time where they just simply give up and stay still. So I'm hoping you all see that data. And this immobility time becomes like almost nothing. And the forced swim test, they'll also continue swimming. They won't just float and give up. So by knocking out P4H, it has some real effects, both on socialization, it has effect on mood, and it also has an effect on fear as well as anxiety. So here's this question for you. Why is this paper only published in neuropharmacology? Why isn't this in like Nature? Is this data not clear? Is this data not novel? Like, why do you think it's published here versus elsewhere? Any ideas?
Does it maybe have to do with knocking out a transmembrane protein, like as a therapeutic approach? One would be kind of difficult to do, and number two, the reduction in anxiety isn't always the best thing, because sometimes that anxiety and fear is there to be like, oh, it's probably not a great idea to jump off a bridge.
Okay, so all valid points. So one, does this actually have a translational therapeutic effect? They actually don't address that in this paper. They kind of hypothesize it, but they don't actually do it. So that could be a limitation. Anything else that you can think of? So what else would you want to know about these animals? Like, is there anything else that you'd want to know about these animals? Like, there's actually one thing that you want to know. Like, if you're thinking about it very, very carefully, does this show that this animal is moving more? It is, right? So it's moving more, it's not immobile, so it's swimming more, suspended more. So this animal is approaching more, right? So is that also related to movement? Is this related to movement? Open arms. It's in the open arms more. So what they're not showing you here is, does this animal actually have increased hyperactivity? And so by knocking out P4H, are you actually inducing hyperactivity in these animals? And you have to kind of think a little bit about why this particular paper, even though the data looks really clear, even though the data is like really, it fits in really nicely, you have to think a little bit about why it's published in this lower ranked journal versus a higher ranked journal. So this is something that you'll have to think about. It's a hard question, and you'll get all of this again in the workshop with Dr. Pressy as well as the other students. But I always want you to be thinking about what you would want to do next. From this type of experiment, what might you have wanted to do next? What are some of the controls? So movement, overall hyperactivity, those types of things, could you have wanted to ask for a control for? And again, always go back to that question, what could they have done better to get this published? And maybe I'll put you on the spot a little bit, Jessica. So if you had like results and they were kind of good results, but you weren't able to get it published, like would you continue to try to get more results and to get a better story and to try to publish it in a higher ranked journal, or do you kind of just go with like the best that you can go with? Like, two different strategies.
I guess it's context dependent for me. So like if I am done my PhD and I just need a paper to defend and leave, and I don't want to continue in research, I'll get whatever paper I can to graduate and I would be done. But if I wanted to get a high impact paper for a postdoc position or something like this, I would continue to look for a mechanism or look for how it altered or featured like a circuit in the brain in order to publish at a higher impact journal. That's what I would do.
So based on what Dr. Pressy just said, is there a mechanism in this paper? There's no link between like alterations in P4H and there is no mechanism. So it's one of those reasons why it is in a lower ranked journal. So there are things for you to always be thinking about. And again, we're kind of here to help you think about those questions and maybe get you to think about what kind of controls might have been done or been missing, as well as other aspects of the paper. Okay, so don't think of it as like, wow, this is a big part of my mark, and I'm really worried about it. We want to be able to help you develop that thought process and to try to help you along as much as possible. Okay, so scientific reports, is that a good journal? What do you think? So neuropharmacology is not a really high impact paper. It's not read by the same groups of individuals that might read Nature or Cell. Like, what do you think of scientific reports? Have you heard of scientific reports before? No, never? Would you want to publish in scientific reports?
Yeah, maybe.
Yeah. So this is sort of one of those journals that isn't like the top ranked journals, but it's considered a relatively good journal. So I know people in CSB publish in scientific reports fairly routinely, and it is kind of a better ranked journal. And I wanted to talk to you a little bit about this because this paper that came out not too long ago is actually a fascinating paper. Their data is not as clean as the data that I showed you, and yet they don't really have a mechanism either, but here the paper is kind of interesting for other reasons. So have you all heard of the microbiome? All of you have, right? And you've all heard of the transplanting of the microbiome. So what do you think that would do? Like if I take the gut microbiome bacteria, mainly bacteria, from a donor, and then I transplanted that into some kind of atypical depressed individual or an individual with autism, do you think that will help? Yes? So do they work in human trials? So most of the things that we read about within the gut microbiome and transfer are done in animal models. This is one of the rare examples where we're looking at human trials. And in these human trials, even though the numbers are small, less than 20, these were pretty amazing results. And I'm going to take you through them, and I'm also going to try to impress upon you the way that this field has changed in just 2 years' time. Okay, so again, there's kind of a lot on this slide. You can take whatever you want from this slide. We know that diet has a huge impact, not just on the human gut microbiome, but also like just in lab animals and in mice as well. And we know that there's usually in mice that there's a balance between two different types of bacteria, the bacteroides and the firmicutes. And this balance, when it gets out of balance, especially after changing the diet, it can actually lead to inflammation, inflammatory markers. It can lead to changes in the permeability of the gut, and all of these things in turn are thought to impact the brain. And so this is relatively new, but it's information that we have been gathering over the last sort of 8 to 10 years, and this has been accelerated over the last 2 to 3 years, and it's been really, really exciting to see all of the potential markers and changes that occur. And we know inflammation, leakiness of the gut, the activation of the immune system, then the effect on the brain has been really, really exciting to kind of watch happen. So again, all sorts of different types of bacteria have been implicated. We also know a lot more of the mechanism than we did 2 years ago. So 2 years ago, we kind of stopped here. So this is where we would have stopped about 2 years ago, and now we know a little bit more of these molecules here. Do you guys remember what these are called?
Short chain fatty acids?
Yeah, these short chain fatty acids, and we know that there are 3 major short chain fatty acids that result from a change in the gut microbiome. And again, we know a lot more of the mechanism. We know how this might interact with alpha-synuclein, and we might know how it might disrupt alpha-synuclein and how it might actually begin in the gut. And again, we didn't know that 2 or 3 years ago, and now we're kind of moving into human territory, beyond sort of the mouse models, and we are able to identify all kinds of interesting bacterial changes and species changes that occur when we change metabolism, diet, when we change immune status, etc. So again, all of these things are leading to these dramatic changes overall in the gut microbiome and the way that we're analyzing it. And the coolest thing is really the development of these synthetic microbiota. So genetically manipulating bacteria, transplanting those back into the gut to produce some of these different factors like more butyrate, etc., has been really amazing to see the development of the microbiome in a genetic sort of context. And it's been really, really cool to kind of look at that. In today's paper, we're going to look at the donor microbiome and how that might impact. Again, it's not in animal trials, it's in human trials. So just to make that clear. And of course, we always wanted to be able to go back and figure out what's actually occurring. So again, we won't go into all of this. Hopefully some of this is a review for you, that we know that the microbiome is always undergoing changes, and we know that there are 3 developmental changes that occur from the time of birth to the time that you're starting to eat solid food, and then into your adulthood. We see these 3 major shifts within the gut microbiome. And there's something wrong in individuals that have autism. In autism, you don't see this shift. The shift that seems to occur in normal, healthy individuals and what we would call neurotypicals, and I'll show you that phrase a little bit later, we don't really see this development in the same way. There's something wrong and there's something missing within this process. And the fact that we can now look at and very, very quickly use genetic sequencing of the gut microbiome has really opened up this field tremendously over the last little while. So here is some of the data from that paper. And before I get into that, what do you guys remember about autism spectrum disorders? Like, what would characterize individuals with autism spectrum disorders? Yes.
I've heard, and I don't know if it's a myth, but when they're that are very young, they feel like you.
So there are definitely social interactions that are not properly developed. So eye contact being one of them, yes.
Sometimes they have difficult with sensory overload.
So sensory overload, intense world theory is like another sort of aspect of that as well. Anything else that you remember?
Trouble reading emotions.
Right, so the ability to sense affect is really impacted in these individuals as well. So the other thing that you probably have heard of is that they are often accompanied by changes in the gastrointestinal system. And if we're looking at some of these changes that were identified in this particular paper, along the x-axis and along the y-axis, you're seeing a number of different types of changes. So on the bottom, we're looking at the gastrointestinal symptom rating scale. So when they have gastric discomfort, along this x-axis, this shows you the overall changes in gastrointestinal discomfort. So they all begin with having a fair amount of this, and then it disappears over time if they are receiving this gut microbiome transfer. And the other thing that we're also looking at is this percent change in different readings like autism rating scales. You'll hear a little bit more about that later on in the course, I think. And if we look, there is a positive correlation between if we do a gut microbiome transfer, there is a positive correlation between the lack of these childhood autism rating scales. So they don't have all of those different deficits that you mentioned before. So we see less and less of them. We see a bigger and bigger change. At the same time that we see a better sort of gut response. So they don't have as many gastrointestinal problems, and behaviorally they're getting better when they've gotten this gut microbiome transfer. And this is all in human studies. And this human study is also unique for 2 other reasons. So this is the first human study where they did fecal transplant, gut microbiome transplant using fecal matter. And then they also followed it 2 years after they stopped. So again, this is a long-term study, very rare to see that. And let me ask you this question. When you do a gut microbiome transfer, what do you remember is important in that process? How do you get rid of the existing gut microbiome? So antibiotics, that's one. So they use very strong antibiotics, vancomycin, to get rid of all of the pre-existing gut microbiome. Then they give another normal, neurotypical gut microbiome from an unaffected individual. How do you make sure that that survives? So when you give someone like a gut microbiome, they swallow a pill. Remember, these individuals have like autism, so you don't want to be injecting them, so they get a pill to swallow. What do you do next? How do you make the bacteria survive longer? Go back to your physiology. What might you do? What kills bacteria in your gut if you swallow something? You guys all took physiology, right? Do you remember? Any wild guesses? Yes.
We can decrease the immune system reaction to the microbiome by
Okay, so in theory, we might have to do that. They didn't in this study. Anything else? Like you just ingested a whole bunch of bacteria. What might you do to allow them to survive longer? Yes.
just to like decrease the acidity of the stomach.
Right. So the modern sort of studies that are now being done on humans, which weren't done like 5 years ago or longer, actually now involve giving drugs to actually prevent the gut acidity. So that helps the gut microbiome to colonize a lot better. So we didn't used to do that. It didn't used to be that important. And so when we saw the initial human trials a while back, a lot of them failed because the gut, the bacteria weren't able to survive within the stomach. But now with these new trials coming out, they actually do. And we can see all of these different changes. Some of them are positively correlated. So the changes in behavior are positively correlated by getting better gut activity. So their gastrointestinal symptoms are getting better and better, and so are their behavioral outcomes. So all of these positive correlations here. And then if we look at some of the negative correlations, things that are associated with bad types of behavior, we actually see a reduction in some of those. And again, I'll come back and talk to you about that in a few moments. So here's a really busy slide, and I don't know, would you present all of the data on this slide? Do you find this a busy slide? It's a really busy slide, and you kind of have to think a little bit about, do I want to present this? So I've only presented a few of the different graphs. They have a lot of them. And do I really need to present all of the different graphs on here? So I'm just going to orient you a little bit on what they were actually doing. So they took a baseline measure of the gut diversity. So as you would expect, because they already have gastrointestinal problems, they don't actually have a lot of gut diversity. The higher on this y-axis, the more diversity, the different types of bacteria that you have. And in the guts of individuals that have autism spectrum disorder and high autism spectrum disorder, there isn't a lot of gut diversity. So 3 weeks after you have done the fecal transplant and after you have done the microbiome transplant, you still don't see a lot of gut diversity. So they've done everything, wiped out the normal gut microbiome, then they've transplanted it, and they've used inhibitors of gastric acid secretion, and it's only until week 10 where they start to see an increase in the diversity. Again, they stop here, and then they go on and track these individuals for changes in autism-like behavior, and this 2-year follow-up where they have done nothing since the end of the treatments here. It turns out that once you have this colonization, it actually is maintained in these individuals. So the gut microbiome stays intact. We get better diversity of the gut microbiome after transplantation, and at this point, they are still behaving much better in terms of the autism spectrum rating scale. They've also shown that there are certain classes of the bacteria that have increased. So bifidobacteria has increased and Prevotella have increased. These are generally found to be favorable in humans. So if you exercise, you generally get more of the bifidobacteria and Prevotella. And they also indicate that they see changes in a different bacteria that decreases. So it's not an overall, everything is going up, some things go up, and some things go down. So again, do I need to explain the entire graph, or could I have gotten away with just like having a few bullet points? I think those are things that you'll learn more about in the workshop next week. So did you have any questions about this? Yeah.
Were they required to follow a specific diet over these 2 years?
So that's a great question. So were these individuals after the end of treatment, were they required to stay on a strict diet? No, they weren't. It's a great question.
Out of curiosity, did age group matter?
In this particular study, it's only 19 individuals that they were able to track over this 2-year period. They had a wide range of ages, so all the way up to about 17, 18 years of age, all the way down to about 5 or 6 years. So there was a wide variety of age ranges, but it's beyond sort of that the 1 to 3 year interval where we know that the gut microbiome should be stabilizing. So another great question. Yes.
I guess you kind of just answered it, but would the donors have been similar in age to the?
No, they were not. So it's one single donor given to all ages. So that's a great question. So could we have gotten even better results or have different results if we tried like age matching? That wasn't done. So it's like one donor microbiome that was transplanted to all of these different 19 individuals. Again, a great question. Yes. No, not so far. I don't think this has been that long since the study has come out, so I don't know if they have a further study, and that's another great question. Like, does it maintain even after another 2 years has gone by? Great question. Anything else? Yes.
So I think that between all the patients, they all have low phylogenetic diversity, so they have low diversity of gut microbiome. Did they, I don't know, is there a reason why it's so low?
Yeah, so it's a great question too. So when you look at autism, and you look at autism spectrum, so here in Toronto, they're looking for genetic causes for like autism, and it's kind of interesting that Stephen Scherer, and I think you work with Stephen, right? Or did you work with Stephen?
Not Stephen Scherer directly, but Benjamin Blanco, who has a lab. Their lab is down at Sick Kids Hospital, and they look at genetic underlying or causes of autism spectrum disorder, and that they're looking at particular mutations in genes or methylation states of genes that increase the severity of autism in different individuals.
Right. With that said, if you look at either neurodevelopmental or you look at even some neurodegenerative conditions, there usually is something beyond sort of either the motor impairment, cognitive impairment, or something that goes wrong with the gastrointestinal tract in a large number or proportion of these individuals. We don't know if it's the cause or if it's just correlated with it. Again, we kind of hope, because that's what they're hoping, is that it is the causal agent, but there is no mechanism here.
I also want to kind of follow that. So it's autism spectrum disorder. So it's a spectrum, right?
Yep.
So does the degree of the diversity?
So wonderful question, and it's a great question. So what you're asking is, does each of these data points map, like so very, very low diversity, mapping with like really, really strong Asperger's, because Asperger's was included in this particular study, versus someone with very mild sort of autism rating, higher diversity. So they weren't able to map individuals in the same way. So that's a great question too. Their numbers are so small, like there are only 19 individuals. Still a lot of data points for them to kind of sift through, but it's a great sort of point. So down the road, when we get into being able to sequence larger numbers of these genomes simultaneously, then that should be answerable. But for this study, no. It's a great question. Any other questions? Yes.
Other than like the gastrointestinal issues, were there like other symptoms, maybe like social symptoms that you could see were changing over the course of the 2 years? Or is that something that they didn't really follow?
No, so they were seeing that after microbiome transplant treatment, that they did indeed see better sort of behavioral outcomes in these individuals, and that their gastrointestinal distress was getting better too. So both of them correlated in a positive way. And that was one of the slides that I showed you earlier.
Okay, yeah.
Anything else? Yes.
So this was in humans. Has anyone kind of tried a similar study in mice to look more at the mechanism, like try clipping maybe the vagus nerve and seeing if that results in, since there's no communication between the gut and the brain, you don't get the same benefits?
Yeah, I think that would be a great question to ask like Dr. Benko. So I'm actually, or someone from his lab at some point, because I think there's a lot of there are a lot of questions around autism spectrum disorders, and whether it's purely genetic and environmental, and whether or not everyone even agrees like on a mouse model. So in some instances, even like agreeing on a model is challenging. So like your ALS model, is everyone using the same one?
No, and there's continuously new models being developed even now. So I know in autism spectrum, every new gene that's being identified, like right up even until today, people are trying to make a mouse model of that. And whether or not those mouse models are being accepted in the field, not necessarily is the case. So yeah, it's just ongoing.
Did you have any other questions? So I think we're just going to wrap things up a little bit early. I know it's lecture one, so it's a little bit it's a little hard to get into the swing of things. So if you want to spend a few minutes, we're going to try to do this as much as we can to give you a little bit of time before it's supposed to be your ticket out the door. So before you leave, if you can finish it, just leave it up here. Please make sure your name is on it. And that you've indicated and finished everything here. I will post your mark for you out of 2.5 tomorrow, and you'll be able to see it directly on Perkis. I'll bring them back with me the following class, so if there are any comments that I need to go over with you, all right? And the other thing, if you do finish early and you leave this up here at the front, start thinking about who you want to work with in the class. If you don't have a partner, then maybe we can start like a discussion thread of people who are looking.
So in addition to this, I'm going to be opening up a section on Perkis where you can put you and your partner's name into Perkis, so we have the partners specifically outlined. So make sure that when you find your partner that you fill that in. It should be up later this week. And again, for anyone who doesn't have a partner in the class, we'll definitely start a discussion board for that and start connecting you into pairs. Okay?
So as Dr. Basti was mentioning, so one of the things that really separates one is that we just publish a lot of observational studies, is having a really the mechanism behind it, interesting there's a number of different isoforms. No, so I think that would still be pretty descriptive. And if you're looking at how might